Assalamu alaikum. This is Iba Mahmood, AP 1770054. The topic assigned to my group for today's presentation is to study the cross-behavioral activities of mice, of which I will be presenting the test for spontaneous activity. Before I begin with my presentation, I just want to say here that I'd like to apologize in advance for any disruption or background music or sound that's happening in my environment because there's a Nathwani that's happening next door. So that's fine. So starting off in the presentation, first of all, there is a brief explanation of the importance and cruciality of cross-behavioral activities itself, as they're used to determine the cognitive function, psychological status, and physical conditions of the rodent that is under observation. And these assessments can further be used to determine the pharmacology, toxicology, and physiology that resides among the animal. The test for spontaneous activity is the extent of free movement by mice in a familiar environment, which directly reflects its physical and mental conditions. So spontaneous activity has its own purpose and significance. Some of the most highlighted ones I've mentioned here. Number one, it provides assessment of gross motor skills. Number two, better used to assess traits such as anxiety, response to novel situations, stimulant activity of drugs, and experimental compounds. Number three, it has also been proposed as one of a battery of behavioral tests for phenotyping genetically altered mice. Okay, so we have two methods for testing spontaneous activity. One is a traditional method, and the other one is rather a contemporary method. The traditional method we have here is non-automated assessment. So the method is very simple. We take a box that has a dimension of 100 by 100 centimeters. The walls are opaque, transparent, and they're high enough to prevent the animal from escaping. We divide the box into equal sections by using a marker pen. The test sessions are usually recorded on videotape to reduce the possible confounding effects of an observer. And the experiment is performed in a quiet room in order to prevent any interference. The procedure is very simple. To perform the test, an animal is placed in the middle of the cage. The observer then records specific behaviors over a prescribed period of time, which is usually between 5 to 15 minutes. Oh, sorry. Yeah. The parameters to record may include roaring, locomotion, grooming, and stereotypical behaviors such as licking or biting. After the data is collected, we have different software packages that can analyze the video recording of animal movements and provide tracings of an animal's path and the distance traveled. So I have a diagram here, and over here, basically, we have a number of activity cages that are placed close together, and we have a rack. On top of the rack, we have a video camera. So the video camera is used to observe the rodents that are under observation. This method is slightly different from the one that we perform in our lab, considering that we have only one activity cage and only one specimen that is under observation. But when we're performing this experiment on a more larger scale, we can have a number of activity cages. Right. So that concludes the process or the method and apparatus of non-automated spontaneous activity assessment. Moving on to the automated assessment, and the apparatus is very compact, and yeah, so it has an activity monitoring system, which includes an analyzer and automated activity box, which is equipped with infrared light beams and photocells to measure horizontal locomotion, and with vertically mounted infrared light beams and photocells to record rearing and jumping. So what happens is that the surface is divided into grids, and there's an x-axis and y-axis, which is used to observe the horizontal locomotion made by the animal, whereas the z-axis is solely used to determine the rearing and jumping movements made by the animal. The method and material is also very simple. The activity boxes range in size from that of a standard clear-sided rodent cage up to approximately 40 by 40 centimeters for mice. This means it can be different for rats or rabbits or whatever specimen you have under observation. The infrared beams are coupled to two sets of photocells and act as motion directors that collect data for both vertical and horizontal movements. The activity box is connected to a personal computer that can produce data from one or more boxes simultaneously. and the data can then be transferred to commercial software programs for further analysis. All right, so the procedure is also very simple and similar to that of the traditional method, as the test protocol is similar to that prescribed for the non-automated spontaneous activity assessment. Prior to beginning the test, the computer is programmed to record the activity. The animal is placed in the test arena. The data is collected automatically over a prescribed period of time. So I have a small diagram here. This shows that the rodent is placed inside the activity cage, and the activity cage, as you can see here, has been divided into equal sections or grids with the help of infrared light beams. The movements, whatever the movement or spontaneous activity made by the mice, is recorded. And then the analysis we make through the different software programs in the computer can then be used to plot our graph. So in my research article, we had, I had 4 graphs here. So this experiment is basically a comparison between WT and KO mice. So KO mice is a knocked out mice, which has a gene removed from its body. Yeah. So it's basically a genetically altered mice. And a WT mice is basically that which is the wild type mice, and it is considered as the normal mice we use at the laboratory. So the first 2 graphs determine the travel distance and resting time made by the 2 mice, whereas the third and fourth determine the walking speed and cage activity made by each of the mice. So a general result that we can produce from these graphs is that the KO mice has as you can see here, it has a greater resting time, whereas the WT mice has a less resting time, which indicates that it has more agile activities. As you can see here, it has more walking speed, it has more cage activities, and it has more travel distance. And one more thing that I want to mention here is that the automated assessment system has different features. This means that it can be changed into the dark conditioned or light conditions, and there are further temperature, humidity, and other external stimuli changes that you can make within the environment, which gives it another edge comparing to the non-assessment system, automated assessment for spontaneous activity. So I will be discussing the advantages of automated over non-automated spontaneous activity assessment. First of all, the direct observation of each test subject can be laborious and inefficient for studies involving large number of animals. It's very obvious that non-automated assessment requires more labor, requires more manual work comparing to the automated assessment of spontaneous activity testing. Number 2, in automated assessment, the computer can be programmed to record data for periods of an hour or more, which is beyond that, which can be made by an observer to accurately measure the mice activity manually using the non-automated system. Number 3, the computer can separate data for activity performed in the center versus peripheral areas of the arena. That means that you can have separate data for the movement made by the mice in the horizontal axis and vertical axis, which gives it another edge. And number 4, the newest generation of activity boxes can control for the effects of light, temperature, sound, and limit influences by external stimuli even further, as I have mentioned earlier. Now, there are certain variations that we can make in the spontaneous activity testing, although the system pretty much is perfect and gives very accurate results, but according to the observer's feasibility, there are certain variations that you can make in it. Number 1, the observer may wish to record activity data for peripheral and central areas of the arena separately, like I mentioned before. Number 2, if the effect of a drug is to be examined, the animal is habituated to the apparatus by exposing it to the activity box several times. This means that if you want to determine the effect of a drug in an animal, then what you're going to do is you're going to expose the animal to the environment it's going to be performing its activity in a couple of times, preferably 3 times for 5 minutes each. And because of that, it will be habituated to its environment. And this habituation leads to a reduction in baseline activity, which gives more precise results. Number 3, habituation of locomotor activity can be used to assess memory in mice. This means that, let's say if I expose my mice to an activity cage. The first time it's going to show more activity, more anxiety maybe, but the next time I re-expose the mice to the same activity cage, it will show less activity. The reason is because the mice has memorized its environment. And this is why we can also use this method to assess memory in mice. The mouse, all right, yeah, okay. Habituation of the apparatus is not performed if anxiety or response to novelty is being studied. Obviously, if you want to check for the anxiety level of the mouse, how much anxiety is going to show when it's exposed to the activity cage, habituation is not preferred. Number 5, a modified open field apparatus that is modified into both well lit and dark areas can be used to assess anxiety. The relative time and activity spent in the two zones then is compared. Anxiolytic drugs can be administered and will generally increase the time animals spend in the well-lit areas. Okay, number 6, it is preferred to perform the test during the night cycle when rodents are more active. Okay. So even though the apparatus we have for the automated assessment is very safe and gives precise results, there are still some animal welfare concerns that arise from this testing. And these concerns are, number 1, if animals are pathogen free, then care must be taken to avoid exposure to other animals or contamination of the testing apparatus. The reason is that if you expose it to other animals, it might get some of its germs or pathogens, and the apparatus itself should be very clean, and the ambient temperature and humidity should be comfortable for the animal, because external stimuli, environmental conditions do have a prominent effect on the behavioral aspects portrayed by the animal or rodent that is under observation. Number 3, animals undergoing behavioral testing often are singly housed, and the addition of environmental enrichment to compensate for single housing of social animals may affect exploratory behaviors in spontaneous activity testing. So that's it for my presentation, and I hope I was able to avidly deliver my portion as properly as possible, and I hope you have a nice day ahead. Thank you very much.